Very much. Welcome. Hi, everybody. You can hear me okay at the back? Yes? Great. Okay, so I'm going to talk about something. I haven't been to the rest of the conference yet, so I'm expecting this is going to be quite different from other talks at the conference. It's going to be a very applied talk about an application I've been involved in of machine learning, Cisco learning, in particular the lasso in medicine. And I think it's quite interesting. I'm excited about the final result, and I hope that you'll be interested as well. I'm a professor not only in statistics, but in biomedical data sciences. So a lot of my work time is spent in collaborative work with people in the medical school at Stanford, which I find very satisfying and very challenging. So as was mentioned, one of the things I'm known for is the lasso, which is basically it's a method for supervised learning. Right? We've got something we're trying to predict, a Y, how long a patient lives, the return of his stock, something like that. We're trying to predict it from a set of features that we know about say that stock or the patient, right? And the lasso fits a linear model. With weights beta j. So the first part of that expression, I'm trying to see if this pointer works. Is there a pointer? Anyway, if someone brings a laser pointer, I'll be happy to use it. Do we have a laser pointer, or is there one? There's one on here. No, he's going to bring me one. Okay. It's okay. There's one right on here. I don't want to advance this. Oh, okay. Thank you. Oh, okay. That one didn't have one. Okay.
I'll take this one.
But don't this advance the slide still? Okay. Okay, so the first part is just least squares regression, right? Which you'll all be familiar with. And the second part is what's really unique to the lasso. You put a budget on the sum of the absolute values of the weights. And this budget is user chosen, usually by cross-validation. And by choosing a small budget, we force the model to be sparse. We force a lot of the coefficients to be zero. Right? So what the lasso says is, well, it tries to fit as well as possible to the data with a constraint on how big the weights can be. And this absolute value constraint sets some of the weights to zero exactly, which is very convenient, both computationally, but also for interpretation. So the lasso goes through your data and says, well, here's the best fit I can find. A whole bunch of the weights are zero, because those features aren't useful, in my view. And for the ones that are useful, it gives a weight, a non-zero weight. Right? So I think, hopefully all of you have seen this before. It's convex, and I guess my good friend Steve Boyd gave a great talk yesterday about convex optimization. I've learned a lot from Steve, especially the fact that convexity is very nice. Okay, it's really a wonderful property to have. It means that you can find the unique solution to a problem like this, and you can do it very quickly. So like, we can solve the world, this kind of problem now for hundreds of thousands of variables. Well, let me say tens of thousands of variables, hundreds of thousands of observations at least, and just on a Mac, on a scalar computer. And if you use a distributed system like H2O, you can solve much bigger problems. So the convexity obviously is a very, very useful property. So we have GLMNet, our lab has written, which is a publicly available software in R. It's also available in Python and MATLAB. H2O.glm in their package also does lasso and lots of other things. So there's lots of different software packages for this, and it's pretty widely used now. So this talk is sort of about the lasso, but it's actually about a very special version of the lasso, as you'll see. So what is this talk about? Well, it's, again, it's a case study. An application I've been involved in, it was just published. How many units of platelets will Stanford Hospital need tomorrow? So a unit of platelet here's some platelets. This is a bag of platelets. It's a bag about 6 inches long. It's an important blood product for clotting, especially for surgeries, therefore. If you give platelets, it's not like giving red blood cells. It takes about an hour and a half sitting in a chair, and the blood's extracted from your bloodstream. The platelets are removed, and the blood's recirculated. It takes about an hour and a half to give a unit of platelets. Right? So people who give platelets are very, very devoted donors. So it's a valuable resource, and I want to talk about how to not waste it. Okay, and my collaborators, Ta is the head of the Stanford Blood Center. These are two of his pathology residents. This is my current student, and this is a grad student of John Taylor, who's recently graduated. And my students especially did a lot of the work in this project, and I'll talk about that along the way. And a good friend, Naris, is going to be a key person in the deployment phase of this, because this is going to produce a software package that implements what we're going to see. Okay, so, and the paper just published in PNAS that summarizes this is given here. Okay, so what is the problem? What am I talking about? The problem is as follows. At Stanford, at least, there's a hospital. It's actually 2 hospitals. And there's a blood center. And the blood center is the sole source of blood for the hospital. Okay? In larger cities, there's typically more than one blood center, but here's one blood center. But the problem I'm talking about is going to be important sort of around the nation. But particular configuration here is there's one blood center supplying blood. And every evening, this hospital sort of contacts the blood center and says, I think we need, say, 40 units of platelets tomorrow, or 35 or 45. And that number is gotten now just basically as a guess. How many surgeries are planned tomorrow? How much blood is on the shelf? It's a guess as to how much they're going to need. Now, here's the real issue. The problem is that platelets have only a 5-day shelf life. Most blood products are good for 40 days or more. These have 5-day shelf life, and the first 2 days are used to test for safety, right? So there's actually 3 days where they can be used, and then they have to be discarded. So there's a limited shelf life. So this is really the challenge. And this is the case everywhere. It's not just at Stanford, but everywhere. You've only got 3 days to use them. So what tends to happen is the hospital tends to overorder, right? They're worried about that they might run short, right? It's a problem because they have to find blood then on short order from some other place. It can be expensive, it can be difficult to find. So they tend to overorder just because they don't want to run short. So currently about 1,400 bags a year are wasted. That's about 8% of the total blood ordered. And that's a really valuable resource, right? It's very hard to get this blood, because I say when you get people to donate it, monetarily, I'll talk later, that's a lot of money too, wasted. There's really shortage, but the problem is there's a lot of waste. Okay? Can we do better based on the available information? So the head of the Stanford Blood Center approached me and said, I've heard about the statistical learning stuff, the supervisor, can you help us to do better? So the data overview, this is sort of what it looks like. This is typically, we have 3 years of data, by the way, historical data. I'm going to do some kind of training on 2 years, and then I'm going to test forward on the 3rd year. The data looks like this, roughly, so you get an idea. Here's the daily consumption. It averages around 40 or so, right? But it's quite a bit of variation. Here's 3 days, cumulative assumption. It averages around 120. But there's a lot of variation, right, day to day. Okay? And there's also variation by day of the week. These are box plots of the daily and 3-day usage by day of week, and I don't know if you can see this, but as you would expect, it's lower on the weekends because there's fewer surgeries on the weekends. Okay. But the main point of these graphs is just there's quite a bit of variation. Okay. So what data do we have? Now this is sort of for people who are sort of getting started in data analysis, this is sort of an eye-opener. You might think Stanford Hospital is a very modern hospital with modern data systems. The data is just there, right? I mean, you think, well, the data is just in a database and let's just get it. No, actually, the data is a mess. There's like the data is in 3 different databases. It's messy. There's a lot of data that's not observed. It's missing for various reasons. My students spent a lot of time just getting the data into a form that was usable. And so this is a surprise that people getting early in data analysis. A lot of the time you're going to spend is just on grunge work, right? But it's important grunge work, because if the data is of low quality, you can't get good answers. So don't be worried that you get this degree in statistics or machine learning, and you spend a lot of time doing what appears to be low-level grunge work, because it's important, and we all do it. And so I give my students really the credit for doing this, because there's a lot of important work. So what data do we have? We have the number of platelets on a given day. So if it's historical data, so we know for the last 3 years how many platelets were actually used on a given day. And from that, we're going to try to predict. That's what we're trying to predict. We have features about the individual patients. For every patient in the hospital, we have their CBC, their blood counts of all kinds, lots of information, census data in each ward of the hospital, oncology, cardiology, hematology, blah, blah. How many patients are there? When were they admitted? What's planned for them? All the information, surgery schedule, lots of information. Of course, there's some things we can't know, like tomorrow there may be a terrible car accident requiring 50 units of blood. So that's not predictable, obviously, but a lot of the information is there to potentially predict things well. But what happens is, as I mentioned, and this is not uncommon, the data is a mess. We tried to work at the individual patient level, but we failed because the information on individual patients was very incomplete. 30% of patients have their CBC not observed at all. And then we can't get an explanation for this yet. It's complicated, it's messy. And lots of other problems. We couldn't match patients with surgery schedules and lots of different things. So we end up basically giving up on the individual patient level and working on the overall hospital level. So we aggregate features over all patients on a given day to make the prediction at the hospital level, because that's actually what we want to do, right? We want to predict for the hospital how much platelets is going to be used, right? It'd be good to do it by patient, but we don't have the information. This is not good enough. So the feature construction then is we aggregated features together. Whoops. For each day I and feature j, we count the number of patients below the first quartile of the population in CBC. Okay? And use the average of the past week. So these are just sort of summary features trying to figure out how many patients have low blood counts. And then the census record, the same kind of thing. The number of patients at location J, maybe these are the wards of the hospital, summed up over different locations. Okay, so these are kind of aggregate features. And more things, the platelet transfusion record for a given day, how many platelets were transfused, surgery schedule, day of the week. 61 features in total. So we have 61 features from which we're trying to predict the blood counts, the blood usage in the next day, and in order to figure out how much to order. Okay, so just some notation. I know it's hard to read notation on a slide. I'll go through it quickly anyway, but if you can absorb it. So this is what we're trying to predict, the YI, the platelet use. We're trying to, the amount of new blood that arrives on a given day, we'll call XI, that's the amount that's ordered, right? And then there's a 3-day shelf life. So on any given day, there's like blood that's 1 day old and blood that's 2 days old that's available to use, right? And then WI is how much we waste, and SI is how much we're short. We want no waste, and we want no shortage, and as little waste as possible, right? I said that at the bottom. Okay, so this is the goal. So our first approach with this high quality graphics I've illustrated, I know sometimes hand drawings are okay. Anyway, is to use supervised learning, right? We've got 3 years of training data, so let's predict on a given day. Let's use the training data for the past k days, and k might be like 400 days, right? So, for example, let's predict forward based on the features how much is used in the next 3 days, right? I mean, train. We can train a model, right? Like a lasso, for example, to do this, and then use this information to figure out how much to order, right? So we're sitting here today on Tuesday. For that predict, we'll need 45 tomorrow and 50 the next day and 35 the next day. I can then use that information by looking at how much is on the shelf and figure out how much to order, right? I'll order enough to make sure I don't run short, probably put in a little margin for error, order an extra 5 bags. But if I can predict well, then I can decide how much to order, right? So this is pretty straightforward, right? Well, so I guess I say that here in more detail. Build a supervised model via the lasso. I tried other things, gradient boosting, random forest, didn't help here. The data is fairly small data, and the lasso seemed to work best for this. So this is just saying in more detail what I said already at the diagram, and we can train either using the past 400 days or 200 days, because things might change sort of seasonally or something, right? So you may want to have the model change as you go along. And then all this is just saying is, well, you use the predictions to figure out how much to order. Basically, this is how much we predict we're going to need. This is how much is left on the shelf. This is how much can we order the last 2 days. So this is just straightforward. So this is one approach. It's sort of the first approach one might think of. It worked pretty well, but the problem is it doesn't really solve the right problem, right? Because if you think about it, the loss function here is not symmetric, right? If you predict too low, it's really bad, right? If this model says we're going to need 25 tomorrow, we actually need 45, it's really bad, right? If it over-predicts, it's not so bad, because you have you waste some blood, which is happening already. So the loss function, the squared error loss function for the lasso is not the right loss function. So we rather pose the problem in a more direct way. Now this is definitely more math than you want to see in a talk, but I've annotated it just so you can get a flavor of it, and you can read this later if you want. But basically, we set this up as a constrained optimization. Remember, the WI is the waste, so we want the total waste is WI, this is over days. Our objective function is minimize the waste, and our predictions are here. So Z is just the sum of the features over 3 days. So we're going to make a prediction, and we're going to put a sparse set of weights on that prediction. And we're going to set up an optimization problem that says minimize the total waste subject to, and all these things are just expressed in the fact that, there's only 2 days of shelf life, so that, there's 1 day old blood and 2 day old blood, and then it's thrown away. But then the last thing is that, here's the big thing, that there's no shortage. So this is the RI is how much remains on day 2. It has to be bigger than 0. Actually, we say it has to be bigger than some C naught, like 5 or 10. So in other words, it says, find me the best set of weights on the features so that when I use the obvious sort of ordering scheme, the number that remain never drops below some number. I'm going to specify like 5 or 10, and the total waste is minimized. Because that's actually the problem we're trying to solve. We're not trying to predict well necessarily the platelet usage. We want a scheme, an ordering scheme that minimizes the waste with no shortage. Okay? So this is, again, a lot to take in on a slide, but if you're interested, you can do it later. But it's just an example of how one has to change the problem to suit the actual problem. This is just an LP. So you can solve it using standard software. And I mentioned a CVXR, which is one of Stephen and students' new products, which is very useful for this. It's basically just an LP. So we specify a lambda by cross-validation, and then we just solve it. It's convex, right? And what do we get? We get a set of weights on the features, and we also get an ordering scheme. We get it says how much should you order, okay? And yeah, so we choose by cross-validation the lambda by sort of a blockwise cross-validation. So what happens? So now we're going to apply this model with 2 years of historical data, and then predict it forward on the 3rd year. Use it to roll forward and ask how well we have done on year 3. First of all, in the lasso does feature selection, the features that were selected were kind of satisfying. The day of the week was a big one. It's kind of a no-brainer, right? Again, because during the week there's more operations than on the weekends. But also things like the number of patients having low hemoglobin, I think this is, or hematocrit, I guess. How many patients were in certain important wards, like hematology and other things like that. So it picked out sort of reasonable looking things, which is another nice feature of this kind of approach, is that the doctor can look at it and say, this is how this is working. This is the basis on which it's making its decisions. Whereas something like deep learning for this problem, or even random forest, are going to be harder to kind of explain, right? This is simple, and here's the weight. We give them weights on these features, right? It chose about 42 features out of 65, I believe. How did it work? Well, I think it's quite successful. Here's the results. Well, first of all, the bottom line is the waste is reduced from 1,400 to 389. So the waste is reduced by, about 3/4, from 8% to 2%. So that's a huge drop. I mean, that's, and here's what it is doing sort of day by day. What do you see here? We see the remaining consumption. Here's the waste. First of all, the remaining never drops below, that's the green, never drops below 0. Matter of fact, never really gets that close, right? Maybe 5. We don't want it to run short. We threw in a buffer, so it looks like it never goes below 5 or so. And the waste is significantly reduced. Now, of course, there's no guarantee this is not going to have a shortage, right? Because again, there could be a plane crash tomorrow or something where we need the Stanford Hospital needs a number of units that was couldn't be predicted. But with high probability, this is going to hopefully do as well as the human system in the sense of not having shortage and having a lot less waste. And then we can choose how much historical data we use. We get even a little better result with a shorter time window, or a longer time window, below 2%. And even, yeah, it's about 2, so it's a 1.5 to 2%. So this is another way to look at the results. Here's under the current system, this is how much is wasted cumulatively. Current system is humans making the ordering. Under the new system, this is how much would have been wasted. So it's quite a substantial decrease, right? This example where there's like low-hanging fruit, like a lot of the work I do, and probably you guys as well, like in say in finance or in genomics, the signal is very small, right? You're trying to get these little 0.5%, 1%. In genetics, you got these very weak SNPs that have to do with phenotypes. Here, the signal is really big, right? I mean, it's low-hanging fruit because what's going to happen tomorrow is largely predictable from what you know today. Not totally, but largely. And this just shows if you sort of organize your information correctly and fit a sensible model to it, you can really exploit that information. So this has got our pathology resident made these calculations that it would save Stanford this much money per year if it was implemented, and nationally $110 million. Again, this problem occurs everywhere, right? So we're deploying it. We've published it now, and this is a Shiny app which NARIS is putting together, and this is going to be implemented in the next month at Stanford at the Blood Center. So it's going to be put there, and the technician every evening will, log into the system, see where the inventory is, how much inventory is there. The system predicts how much is going to be needed based on the current data and the trained model. And we'll have we'll say you need to order 45 plus or minus 5 units. If you do that, we think the shortage will not be more than, not be less. The inventory won't drop below, 6 or something. And the idea also is now to distribute this around the country. This will be a free Shiny app in R, so that a blood center in other place can take the data and can train. They'll train on their own data, because every blood center, every hospital has their own sort of, their own patient populations. The doctors have their own ways of doing things. So you have to train it locally, and then it can be used in other blood centers. So at Stanford, this is going to be used. Actually, it'll be ready in about a month to be implemented, and then my collaborators are very excited. I'm not actually holding them back. They want to actually just put it into the blood center and just start using it. And I'm telling them, actually, no. I mean, they have more of a faith in machine learning than I do. I said, put it in there, run them parallel with the humans for maybe 6 months. Don't let it make any decisions. Just sort of play what if for 6 months. And after 6 months, if it's working as we think it should, then I'm happy for you to put it into action. Because if it goes in the first week, it makes a prediction, and all of a sudden the hospital runs short, that's going to be the end of it, right? Someone's going to say, oh, these machines are so, I never trusted them. We're going back to the humans. So you have to be careful how you make the transition from humans to machines, right? You do it carefully, and I want to make sure it's done here. But I think they're excited about the potential, and also potential for other things in the hospital which have a limited shelf life. There's lots of things which are ordered which they expire after some number of days. And right now there's a lot of waste that can be potentially handled by this kind of procedure. And I also make a plug then, so because I'll have some time for questions, for CVXR. So NARIS, who I had pictured early on, and Steve Boyd, who you probably heard from yesterday, and Angie, a student, they put together something called CVXR. So CVX is a package that Stevens and group have distributed for years, which is a very nice package for convex programming. And I think it started off in MATLAB, and then there was a Python implementation, and now there's an R implementation called CVXR. It's on CRAN and GitHub. And basically, in R, you can just write an R expression that you want to optimize, and you just and CVX runs under the hood. I think it's written in C, I believe. And I found it very useful. And we may well use it for this particular application because we want to do the LP, solve the LP in R. So this is a very nice thing to know about. It's very new. It's only been around for about a week, I think. But you'll find it on CRAN. There's a paper, and I don't know if Steve mentioned it yesterday, but I'll mention it again in any case because it's good to know about. So I'll just go back to this slide and happy to take questions. Thank you.
Yeah, we've got a number of questions queued up on Slido, so once we get it on the screen,
you want me just to read them off?
Well, we can go one by one. So they're ordered. They're ordered by likes. So the first one is can the lasso select variables for GBM?
This is like a quiz or something. Okay, it's
payback, Professor.
How many do I have to get right to pass? Is it like?
You can pass anything you want.
Oh, okay.
So
No,
you can select whatever you want to answer.
Select variables for GBM. So I guess the question is suggesting that you might run lasso first to select variables and then send the selected variables into GBM. You can do that, but not a good idea because the lasso selects variables for a particular model, a linear model. You might have a variable, for example, which on its own isn't very strong, but the interaction of that variable with some other variable is really strong. So GBM would like to see that variable, but lasso would filter it out. So basically, you should do selection of variables within a procedure that's going to use the list. So the lasso does variable selection, which is particularly good for lasso. If you want to do variable selection in GBM, you have to do something which is more tailored to GBM, because GBM, for example, is fitting a different model. It's with interactions. Okay, so can we look at it as a time series? Whoops, rather than a regression. I guess the question is referring to what I just showed. Yep. Time series forecasting. Well, it is a time series forecasting, and I'm doing that, but I'm doing it with a series of regression models, sort of. I'm fitting it at any one time. I'm taking the available data, and I'm predicting forward. So it is, in essence, a time series problem that's been treated cross-sectionally. How do you select the best models when minimizing max residuals is more important than the max residuals? You select the best model. Again, you have to read this. There's an objective function I wrote down which expresses the problem. So you select the model that gives us the lowest objective function. So the objective function was expressing what we're trying to do here, that we're trying to have no shortage and minimize waste. So that's basically the answer is just to optimize the objective function. That's how you select the best model. We choose the model that produces the minimum objective function. Isn't doctor surgery schedule a more accurate predictor here? Did I miss one?
No, they change in real time for whatever becomes the top one. So
this isn't fair. Come on.
Let's answer the doctor one.
Okay, doctor one. Doctor more accurate predictor here. Doctor surgery schedule. Well, we have the surgery schedule, but we have a lot of other information. I mean, a more accurate predictor. It's not a matter of either or. We have the doctor surgery schedule as well as a lot of other information. So it uses that as it can, and then other stuff. So a doctor surgery schedule was a factor that we chose, but a lot of other information was used too. With deep learning methods such as LSTM, I don't know what LSTM is.
Long short-term memory. It's a...
Okay. I didn't try deep learning here because the data is pretty small. I mean, 800 observations, 60 variables. As I mentioned, I tried GBM and random forest, and they didn't help. So the answer is I don't know, but I don't think so. Okay. What if the data becomes non-stationary? Well, it is non-stationary, and that's why we're fitting a model like. That's why our model is actually what we're going to deploy is going to refit the model every week. So the model every week is going to take all the data. Could refit every day, but we're going to refit every week using sort of some back window of maybe 100 days. So it is non-stationary. Good point. Well, we're going to take care of that. How do you determine the minimum remaining bags? Well, it's basically it's a comfort level. So that's something that's going to be chosen by the hospital. Right now, we're just determining, I think it's 5 or 10. Do you have to include blood types in your model? Blood types? I don't think so. It's a good question. Someone knows more about blood than I do might be able to. Sorry. Yeah. You probably have to match blood types when you do a platelet transfusion, I'm not sure. So you need 150 the max, but you recommend 45 if the demand is suddenly up. There could be a problem as the demand is suddenly up. As I mentioned, if there's a huge demand tomorrow that we can't anticipate due to like a kind of car accident or plane crash, there's a big problem. But we're not psychics, so right? I mean, yes. How easy or hard was it to deploy the Shiny app? I'm not doing that part, but at other facilities. Oh, we don't know yet because we haven't done it. Right now we're doing it just at Stanford. That could be a challenge because the data has to have to be in a sort of a reasonable form that looks like the Stanford data. So we're thinking about, we're going to talk to some other centers and see what's involved, but that could be hard. Are the slides going to be able to download? Sure.
Yes.
Yes, that's an easy one. Was there enough shortage to worry about censored observations of demand? Censored observations of demand. Oh, that just went away. Okay.
Well, because people are liking it differently, but here I'll highlight it for you.
Oh, that's okay. I'm not sure what was asked there. Censored observations of demand.
If you ran out of platelets,
Yeah.
you wouldn't observe potential for additional
Oh, I see.
need for
Good,
platelets.
good point. Yeah.
Yeah.
Yeah. So I see. Yes. Okay. I think the shortages are very rare, so there's not enough shortages to have that information. What's the cost per unit bag of platelets? Well, I said something like 1,000 bags a year. So 350,000 divided by 1,000, I think it's about, what is that, 350? So I'm guessing it's about $300 a bag. I have no idea where that number comes from, but that's what I'm told. Linear model, what's the, Lasso predict, linear model of Lasso is better for making less training data. Shall we use random forest if lots of data? Yeah, if it had a lot more data and a lot more features, I think random forest could well have done better. Yeah. Can Lasso predict and buffer extraordinary and unusual patterns? One-time demand, adaptive for the long term. Patterns. I don't know. That's a good question. If there's some unusual patterns that repeat, maybe that could be learned somehow. Yes.
Maybe if the history was long enough.
Yeah. Were more traditional time series methods attempted? Well, most traditional time series methods are trying to model sort of correlations in a time series, right? That wasn't the problem here. The problem really was prediction, so that's why it was more of a regression problem. Is that?
Yeah, well, we definitely appreciate Professor Tibshirani entertaining all the speed questioning here. I think he did a great job.